Okay, so after Alfio's talk, the level of excitement had to calm down. So, so now it's my great pleasure to welcome Esther Pueyo. So Esther, she's from Zaragoza. She's associate professor at the University of Zaragoza, and so she has been doing research in Oxford in her postdoctoral time. And she has been granted an ERC personal grant from the European Research Council, and so she will explain the research she has developed in order to have this grant, and the research that she's doing during its grant about some multi-scale explorations. Of cardiac disease from the ion channels to cell membrane and then so to the upper scale, so to phenotypics considerations. So, Esther, give you the words.
Well, thank you very much, Jerome, for your introduction, and thank you very much for inviting me. It's a pleasure to be here today. Well, I'll talk about understanding heart function, and in particular, I'll focus on electrical heart function, which is the focus of our research, through combined clinical, experimental, and computational research. Well, we all know that cardiovascular diseases are the first cause of mortality, and for these cardiovascular diseases, age is a major risk factor, if not the most important risk factor. And in particular, we are very concerned about investigating arrhythmias, and aging is a major factor for cardiac arrhythmias, and we'll focus on this in the presentation. So we'll start with the electrocardiogram, which I'm sure you all have heard of, which is the electrical signal that is most commonly used in the cardiology practice for diagnosis, for prognosis, or for evaluation of therapy. We see here this signal, for those of you who are not so familiar with the signal. This is the 12-lead electrocardiogram, which you can see here. And you see that these waves, which are characteristics of this electrocardiogram, which is recorded here by using these electrodes on the body surface. Well, this signal is what we see on the torso as coming from the electrical activity of all these different regions in the heart. So the electrical impulse, which generates here in the sinoatrial node, propagates through the atria, the ventricles, and then we see all these waves in the electrocardiogram. Well, this electrocardiographic signal is very commonly used in the clinical practice, but most instances we don't know how to search for really useful information in this signal. So in order to improve the way we use this signal, we can investigate this multi-scale process and see the electrocardiographic signal as coming from a result of all these multi-scale process. So basically we have this electrocardiographic signal here, which comes from measuring the difference in voltage between what is recorded by two different electrodes. Of course, in order to have this electrocardiographic signal in the torso, we need a source that is producing this electrical field, which is the heart. We can go to the level of the heart, as here, and we can measure the activity by electrodes on the surface of the heart. We would have those called electrograms. And over here, in this picture that we can see here, we have an animal heart, and we can measure different signals that we plot here, showing the electrical response of the heart. We can even go one step below and take a piece of tissue within the heart. So we would see here a piece of tissue, and we can evaluate different characteristics regarding the electrical activity of this piece of tissue. The activation, the velocity, or the duration of, for instance, the repolarization, the time it takes the electrical response to go back to the resting state. And we can even go one step below. of course, this tissue is formed by different cells that are well coupled together, and we can look at the activity of one individual cell, like here. We can measure the difference in voltage between the interior and the exterior of the cell, and we have this signal, which is the action potential, which measures the electrical activity of one individual cell. And of course, for this activity to happen, for the cell to have that electrical response, this needs electrical currents flowing through the membrane of the cell. So basically, calcium, sodium, and potassium ions going between the inside and the outside of the cell through these structures, which are the ion channels. And we see here some examples of ionic currents related to different types of ion channels within the cell membrane. Well, if we want to investigate arrhythmias, as is the case of my research, which is very focused on the investigation of arrhythmias, or if we want, for instance, to investigate the effects of therapy, for example, pharmacological therapies, we need to understand all these multi-scale processes. Any alteration in any of these scales, at the ion channel, cell, tissue, or even the whole organ, can lead to alterations that we can see in the electrical signal that we measure in the torso. Well, as you can imagine, there is multiple feedback between the different levels, which makes the problem highly complicated. Also, there is very much variability, both inter-individual variability. We all know that two different hearts from two different people behave in a different way, but also importantly, we have intra-individual variability. So if we see two different cells of the same heart, even if they are neighboring cells, they behave differently. And also there is variability in the temporal domain. So the same cell paced at the same frequency, it shows different responses from one beat to the next. So there is high variability that we should take into account when analyzing the electrical signal. And also, if we want to do some simulations to better understand this electrical behavior, we need to take into account that depending on the problem we want to solve, we may need to account for a large temporal scale, but also a large spatial scale. So in the temporal domain, we go from events that take microns of seconds in occurring, and others that take minutes. And also in the spatial scale, if we want to go from the level of the gene or the ion channels up to the level of the body, we have different orders of magnitude. And here is where modeling and simulation comes to improve our understanding, or to help to better understand what is happening with the electrophysiology of the heart. So it may be seen as a complement to experimental and clinical investigations, where it's proven very useful. But also, it may help into the research of conditions that sometimes are not easy to test experimentally, or even impossible to test experimentally. So we can make use of these computational models to help us research into those conditions. For instance, they are very useful for the investigation of arrhythmias, as we'll see later, or for predicting therapy effects. In this regard, well, I'm sure you know what arrhythmias are. They are defined as alterations in the cardiac rhythm. And you see here, on the left, you see here a heart in sinus rhythm, which means it is beating normally, and so it can efficiently pump blood to the rest of the body. While on the right, you see an arrhythmia, a very dangerous arrhythmia, which is ventricular fibrillation, in which the ventricles quiver and do not allow the efficient pumping of the blood to the rest of the body. And eventually this, if it is not stopped, it would lead to sudden cardiac death of the person in a question of minutes. So if we understand the different manifestations at different scales, we may improve our understanding of why this arrhythmia is occurring, and maybe develop methods to prevent them. So here we see an example of an alteration in a specific ion channel, which is the HERC channel encoding for the IKR current, and we see how these alterations lead to abnormalities at the cellular level, which then translate into abnormalities in the ECG, in the electrocardiographic signals, and eventually could lead to this dangerous arrhythmia. But also, as I was saying before, when evaluating drug effects, this picture here corresponds to the new initiative, SIPA, which is promoted by different regulatory agencies like the FDA. And in these steps to analyze the effect of a compound, the in silico models are seen as an important, a fundamental part for the evaluation. So we see how these models have come into this practice. Well, of course, you know about computational models, which are date back to 1952 for the first neuronal model, and then to 1960 when Dennis Nobel in Oxford developed the first cardiac action potential model, and now we have models for the different parts or regions of the heart. This is just a picture of how an electrical cell model basically works. So basically we have here the cell membrane with these structures, which are the ion channels, apart from pumps and exchangers. And we can see this as model. We can model this as an electrical circuit where we have the cell membrane behaving as a capacitor, and then we have these resistances, which corresponds to the different ion channels that we have in the cell membrane. So basically, we have these ordinary differential equations to define the behavior of the cell. And in these sets of ordinary differential equations, we have here equations for the gates of the different channels. So basically, this would be one specific ionic current, and we model this ionic current through different gates, which allow to model how the ions move from the inside to the outside of the cell, or vice versa. And we have these differential equations to define the gates of the channels. And altogether, we have a set of differential equations to define the electrical activity of the cell. If we want to model how is the propagation in the tissue, when the electrical stimuli go from one cell to the next, we can use this monodomain model, basically one partial differential equation, or the bidomain model with two different partial differential equations together. Of course, I will not talk about this, but we need numerical methods to solve all these big problems. And if we want to derive the ECG, what we can do is to allocate the heart or the ventricle, whatever we are modeling, inside a torso and simulate what would be the activity that we would see at the body surface. So simulating ECG-like signals that we can then compare with the electrocardiographic recordings that we have in the clinical practice. So this was a general overview of what we can use models for. And now I'll be talking about a specific project I'm working on, which is a project funded by the European Research Council, which deals with the investigation of cardiac aging. As I was saying before, aging is the major risk factor for cardiovascular diseases. So if we understand how aging is affecting the heart, and in particular the ventricles, which is the part I'm working on, we will better understand why aging leads to diseases, and in particular cardiovascular diseases. So what are the objectives of this project? Well, the first objective is to evaluate the effects of aging on the human heart, and in particular in the electrophysiology of the ventricles. Once we have characterized these effects of aging, we want to assess inter-individual differences. So we know that two different hearts do not work in the same way, and even two hearts from two people of the same age, what we would call chronological age, are different, are evolving different in time. So we want to understand why these inter-individual differences occur and relate them to the biological age of the heart rather than to the chronological age, because maybe with the biological age we can better understand this different behavior. And finally, we want to unravel the mechanisms that are underlying these inter-individual differences and the relation to biological age, and use those mechanisms to then propose novel risk markers that could eventually help in the clinical practice. So we use an integrative methodology that combines in vitro investigations, also the analysis of the electrocardiograms, which are recorded from the patients, and then in silico models to integrate all these data that we get from the experiments and from the clinics. We'll follow a multi-scale approach, going from the cellular level or even subcellular level to the tissue, we'll do some simulations in a geometry, in a cardiac or ventricular geometry, and then we'll go to simulate ECG signals. We'll consider different factors, mainly mainly electrical factors, because we are focusing on the electrical activity of the heart, but also we'll touch some structural factors, and importantly, the modulation of the electrical activity of the heart by the autonomic nervous system. Also, rather than considering only the effects at the population level, so eventually how aging affects generally the population, we want to understand what I was mentioning before, inter-individual differences. And we'll try to do the effort to go to human. So we've done several studies on animals because we have a more controlled situation and we can optimize all the techniques, but eventually we'd like to do everything in human. So basically, in a graphic, what the methodology is about, we consider a population, a large population of different individuals. We do several analyses at the subcellular, cell, tissue, and body surface level. With this collected data at the subcellular, cell, and tissue levels, we do different in vitro experiments, and from the data that we get from the experiments, we integrate them into in silico models that we use to run simulations and compute ECG signals. We do some evaluations in these signals and compare with the same ECG signals recorded from the same patients from which we acquire all this data. So with this, with all this framework, we plan to investigate, to characterize the effects of aging, and then eventually use them to predict arrhythmia. And this is work in progress because we've started with a project, but there are several years still until we finish with these investigations. So basically, for the human data, we collect the data at 2 different hospitals in Spain. One is Miguel Servet Hospital in Zaragoza and another hospital in Malaga, from where we get signals and data as well. Also for the animal experiments, which we have started with, we collaborate with the School of Veterinary Science, with another biomedical research center in Aragon, and also with data from Merca Zaragoza, where we get data from pigs, sheep, and mouse. Well, as I was saying, this project about characterization of aging effects on the heart has 2 parts. One is experimentation and clinical data, and the other one is computation. So starting with the first one, which is about experimentation, what we do is to take, as a first instance, to take cardiac biopsies. So basically, we take a population of patients which go to cardiac surgery because reasons not affecting the part of the ventricle we are investigating. And from those patients, before the surgery, we take a true cut biopsy, which means a biopsy of the whole ventricular wall. So it's a tiny piece of tissue, and from that biopsy, what we do is to do several analyses. So basically, one part of the biopsy is for cardiomyocyte isolation, so that we can perform experiments in the cells with the patch clamp technique to measure electrophysiological information. But also from another part of the biopsy, we do tissue slices, and we evaluate the electrophysiology in those tissue slices by using optical mapping, both measuring simultaneously the action potentials and the calcium transients. And a third part of the biopsy that we get at this first hospital is we preserve them in paraffin to do immunohistochemistry analysis to characterize tissue alterations regarding, for instance, fibrosis, senescence markers to characterize the biological age of the tissue, and coupling between the cells through these connexins. Well, here's an example of the patch clamp, how the patch clamp technique can be used on these myocytes isolated from the tissues to characterize the currents and eventually also to characterize the action potential. And in the tissues, what we do is to get, to cut the biopsy into these fine myocardial slices, which we can see here, and we simultaneously measure voltage here and calcium, which is not shown here. And these are 2 very small tissue slices that we get from the patients, from the biopsies, and how we can measure the voltage in different points throughout the slice. And as I was saying, we use immunohistochemistry to characterize fibrosis in the tissue, the level of fibrosis in this piece of tissue, the level of cell-to-cell coupling, and also some senescence markers, which are well established in the literature, quantifying different characteristics, beta-galactosidase or P16 as another biological marker. From the same set of patients in this first hospital, we also get blood samples, which we use to quantify plasma markers, and also we get 12 lead ECGs, which we use to evaluate different markers, particularly related to repolarization of the ventricles. And then we get the same tissue samples from another hospital in Spain. This hospital is not in the same city where we have the lab, so we need to freeze these biopsies. And then we use these biopsies, which have been frozen, in order to do different DNA, mRNA, and protein analysis from these biopsies. And from the same patients in this second hospital, we also get blood samples and ECG signals, which we use to do further analysis about mRNA and microRNA, but also to evaluate risk markers in the ECG. Well, this is some work that we have started, this microRNA analysis. Our idea is that we can establish a relationship between microRNA levels in the plasma and in the tissue in order to have some idea of how potentially circulating plasma markers could be indicative of alterations occurring at the level of the ventricle. Of course, this only occurs for very specific markers, but we are working towards understanding that. Of course, this is part of the work that we have started using another database study that we used as a first step, and then we are doing the experimental validation in our tissues. And just as a final part of the collected data that we use, we've started considering that maybe the physical activity was a very important variable in understanding how different persons age. So we've taken volunteers from different ages, and we have collected ECGs and plasma to correlate the plasma markers with the ECG in relation to both age and physical status. Of course, this work will be done after we have performed the work on the biopsies, because we cannot search for plasma markers in any way. We need to base it on the data that we have from the biopsies, where we can correlate blood data with tissue data. And as I was saying before, all this experimental data and clinical data that we get from the subjects and the patients, we integrate them into computational models. And these computational models, we start with the level of the cell. We see here with these differential equations defining the cellular activity. But rather than considering a deterministic model, that is the standard way of using the models in the literature, we are very concerned about the variability, including the variability in the models. So you see here temporal variability. This is the same cardiomyocyte, and this is voltage data. And we can see, well, it's not evident here, but we could quantify differences from one bit to the next. So the same cardiomyocyte has different responses a long time. But importantly, if we take two different cardiomyocytes, even if they are very close together, we see that they have, at the same time, they have different cellular responses, even if they are stimulated exactly in the same way. So we want to incorporate both this temporal variability in the models and this spatial variability in the model. So what we do is to go from deterministic to stochastic models. So basically, we consider the random gating of the ionic gates, which experimentally is well known, that produces fluctuations in the currents, and this leads to differences in the action potentials from one bit to the next. And also we consider that different cells may have different proportion of ion channels of each specific ionic current. So we incorporate that in the models as well. Well, basically, I won't go into detail into this, but basically, if we have one channel with two states, just to start with, we have this probability of transitioning from one state to the other, and we can compute the probability of the channel being open. We would have an equation like this. if rather than having just one channel, we would have n channels, we had n channels, then we would have this evolution of the probability for each of the potential number of channels in a given state. Here we would have for a very large number of channels the expression in terms of proportion of channels in each state, and doing this, an approximation, we would have an equation like this. And basically, in the end, we would have, if we wanted to describe the process trajectories, we would have an equation like this. So this is an stochastic equation where we take into account these different, this stochastic gating that leads to fluctuations in the currents and eventually in the action potential. And of course, this is then generalized to channel models with different multiple states. So if we have an equation like this, as I was saying before, where this is a vector representing the proportion of channels in each possible state of an ion current, eventually open and close if only two channels, if only two states, or many more states, depending on how complex the model is. Well, we have applied different methods, because if you solve this, you may this is representing a proportion of channels, but this may go outside the physiological boundaries that are representing 0 and 1 as all channels being open and closed. So we use these reflected stochastic differential equations to account for that when solving the equation. So what is the idea? Well, we want to use those stochastic models, but what about if we have a cell, a myocyte, and we have recordings, voltage recording and action potentials recorded in that particular cell? How do we know what model we need to use? Because we want to use a model with stochasticity, but with different numbers of channels for the different ionic currents. How do we do that? Well, what we do is we have these experimental measures for a given cell, and we want to estimate both the parameters of the model, meaning the different conductances associated with different number of channels in the model, and also the state variables of the model, which are the intracellular concentrations and the ionic gates, and of course, the voltage. Well, what we do is to formulate the problem as a state space with a state space representation, where we have here the observed data, the experimental data that we get from that particular cell, and we express it as a function of all these hidden state variables, the variables of the model, plus here, when we describe the evolution of these variables, we have here the vector with the parameters of the model, the different number of channels associated with each current. So we use statistical estimation methods. We've started with the unscented Kalman filter for this nonlinear problem, but we are also investigating now methods based on particle filters. And we use these to estimate the specific characteristics for each individual cell that we investigate. So basically, we have this noisy data from the experimental recordings. We use this type of unscented Kalman filter that I was mentioning before, and we get, as a result, we get an estimation of the action potential without having eliminated the noise, and the hidden states plus the parameters of the model. So basically, now we go back to our cell model that we are using as a basis, and we get a reproduction of this data that should eventually represent the experimental data that we had in the beginning. So here we have an example. This was synthetic data that we used to corroborate how the model, how the method that we developed was working. We have the noisy data, then the filter data, and we estimate here as one of the ionic gates in the model, so one of the state variables of the model, and one of the parameters of the model that had this value, and we have this method converting to the value that was in the synthetic data that we used as an input. Here we see how we do it in terms of estimating the different parameters of the models. You see that all of them can be adequately estimated except for the IKS current, which is a particularly difficult current in the models because it has very low impact on the action potential, at least with this human ventricular model, the O'Hara model that we are using. So if we compare the input data, the histogram for the action potential duration in the input data and in the estimated data, we can estimate them very well, and also in terms of the variability. So if we quantify bit-to-bit variability, we can do it well with this method too. If we want to reproduce the experimental AP traces, well, the above panels are for synthetic data generated with different human ventricular models and how we can estimate them with our method, and we see that we can approximate them reasonably well. And here the same for this other model. And these two are two traces of different cells, experimental data that we get as an input, and how we estimate them with our developed model, which can follow this AP trace. And not only the AP trace, but also the variability that is in the model is well reproduced with this method as compared to using a standard human ventricular model, which produces this response rather than representing the behavior of the individual cell. So this was the method. We should now have a method to characterize how is the electrophysiology of different cells and tissues well, eventually tissues when we couple the cells together. But what about the autonomic modulation? Is it important to consider the autonomic modulation of the electrical activity of the heart? Well, it is important, but particularly when we are investigating aging, it is very important because it is well known that one of the effects of aging is to alter the autonomic response. And we know that ventricular cells do not respond to autonomic stimuli in the same way when they are young or when they are aged. So we wanted to introduce these effects in the models as well. So basically, we consider beta-adrenergic signaling model, and we quantify the fraction of phosphorylated and non-phosphorylated channels, and we compute the current as the weighted average of both of them. And of course, in the end, we want to quantify how this autonomic modulation alters the spatiotemporal variability in response to any stimuli. We use these models incorporating the autonomic modulation to confirm, well, to investigate a situation that is occurring in ventricular APD data that we get from patients. This is not for the aging investigations. It was made for heart failure patients. We see these oscillations in the action potential duration that occur with a frequency, with a low frequency. Basically, if we represent here the frequency versus time, what we see here is that there is this, at this low frequency, there is this modulation that is maintained when we measure different recordings of the same patient. So we wanted to use the models with this autonomic modulation incorporated to investigate these oscillations. And we coupled the electrophysiology, the beta-adrenergic signaling, and for these models, we incorporated the mechanics as well at the cellular level, which we did in this investigation. We are now moving to tissue, but for these results that I'll be showing, it was cellular models. So basically, what we got from that study was that these oscillations in the action potential, we can look at this black graphic here, which means how the APD evolves as a function of time. And we corroborated that these oscillations could be produced in the magnitude that we quantified, were produced by both the response to a beta-adrenergic stimulation, simulating this phasic response as in the experimental data that we get from the sympathetic activity, and also produced by this modulation occurring at the level of stretch, mechanical stretch. So both mechanical effects and adrenergic effects were responsible. We checked that both of them contributed to this oscillation. So here is the effect of the beta-adrenergic stimulation, of the stretch, and of the combination of the two. Well, these other graphics that you see here in color are when we include stretch-activated channels as well. And importantly, what we saw is that these oscillations in the action potential duration occurred in control, but also occurred when we simulated different disease conditions like calcium overload or reduced repolarization reserve. So basically, when we combine the two conditions, we observed that the oscillations were amplified, and in some cases, we could see that they led to proarrhythmic events in the form of early after depolarizations or spontaneous action potentials. Well, once we have the models, the cellular models, both at control and also including the autonomic stimulation, we want to introduce the effects at the tissue level. So at the tissue level, we want to introduce both representation of fibrosis and representation of cell-to-cell coupling. And this is work that we are currently doing, so we don't have results for this yet. But we are introducing these different levels of fibrosis and these different levels of cell-to-cell coupling. We are incorporating them into the models with the idea of correlating how tissue alterations relate to chronological or, more importantly, to biological age. So this is the idea of the work that we plan to do, but we don't have results about this yet. And the last step will be to integrate those models, well, build models that we can integrate and compute ECG-like signals. The idea of using these ECG signals is to evaluate different markers that could potentially be markers of arrhythmic risk. So basically, to characterize different patterns of aging for different subjects and having different patterns of aging, which of those patterns is more pro-arrhythmic. So we know that aging is related to arrhythmias, but which are very prevalent in the aging population, but we know that different people of the same age do not have the same risk, are not at the same risk. So we want to understand whether from the ECG signals, which is the signal that the cardiologists see in the practice, we want to see whether there is any sign that can be predicted, or that can be something that we can use as a marker of risk. And this is the work that we are doing. Since we don't have the data yet, because this is a project that has started, but it still has some years of running, what we've done is quantify markers and evaluate them on other databases where we have clinical follow-up. So one of the markers, of the ECG markers that we have evaluated, is this marker that quantifies morphological differences in the T wave, in the repolarization of the ECG. So basically, we have different T waves of the same patient recorded at different heart rates. And what we do is, well, first align the T waves and then compute a transformation from one of the T waves to the other T wave in such a way that it closely it matches the other wave according to a specific criterion, so minimizing an objective function. So basically, this is so-called the warping function, and what we get from this signal is how we need to stretch one of the waves to better match the other wave. And from this information, we can get a variation level in the temporal domain, but also a variation level, a difference in the amplitude domain. So we have used those markers of the amplitude and temporal variation to evaluate these ECGs of patients, chronic heart failure patients, some of which died of sudden cardiac death and others who died of pump failure death, and were followed up for a mean of 44 months. So what we got is what you see here. The top panel is for sudden cardiac death, and the low panel is for pump failure death. So if we use the clinical data, as the cardiologists have in their practice, what we could do is to separate the high and the low risk groups with this difference. So not much difference in terms of separation according to the risk of dying from sudden cardiac death. However, if we use the ECG markers, and in particular the two markers I was mentioning before, we get a much higher separation between the low risk and the high risk. So we can better identify patients that are at high risk. And if we combine the clinical and the ECG variables, we are doing more or less the same as we did with the ECG markers. In terms of pump failure death, so basically death due to failure of the heart as a pump, what we see is that if we use the clinical variables, we already have a quite good enough separation between the low and the high risk groups, similar to what we get when we use the ECG markers, and when we combine both of them, we get basically the same behavior as with the ECG and with the clinical variables. So basically, the markers we are using are very useful to identify arrhythmic risk, ventricular arrhythmic risk leading to sudden cardiac death. We plan to use for the aging investigations other markers that we have already used in other studies, like quantification of T-wave alternance, or which is this alternation in the pattern of the repolarization in the ECG, but also the markers related to how the QT interval in the ECG follows the pattern of change in the heart rate, or other depolarization markers, or ventricular heterogeneity markers. And just to end up with the presentation, I'll conclude by just mentioning another work that we are doing as let's say, separated part of the main project, which is this in vitro study of cardiac aging. So basically, what we are doing now with the patients, we get the biopsies, we get the ECG signals and the blood samples, but we don't have a longitudinal study where we can have different data from the same patients. Of course, this is not possible to do it with the patients because they won't go back to surgery, or we won't have any other data apart from non-invasive data. So what we are doing now is in a study, well, it's done by another researcher in my group, is use this in vitro model where we use genetic engineering to induce aging in a controlled manner using iPS-derived cardiomyocytes. So basically, by introducing the progeria gene, we generate this in vitro model of aging, and we investigate changes in the cardiomyocytes associated with aging, so as to have an idea of how aging would affect the electrophysiology of the ventricular cells without any other external factors apart from those that we are simulating here, well, we are inducing here. So just to conclude, I'd like to conclude by saying that combined clinical, experimental, and computational research, we believe that is key to understand cardiac electrical activity, in our case to characterize the effects of aging, but of course to characterize other cardiac conditions or pathology in different scales from subcellular level to the ECG. Also, it's very relevant for identifying risk of suffering from cardiac arrhythmias, to understand the mechanisms that underlie this enhanced arrhythmic risk in each of the conditions, aging in our project, but other pathologies in other projects, and also to facilitate assessment of clinical and therapeutic interventions and improve the optimal treatment selection. And I thank you for your attention, and I'd like you to invite to come to Zaragoza for the VPH meeting that we are organizing in September. Thank you very much.
So thank you very much, Esther, for this very nice talk. Yeah, question here.
Hello, thank you very much for this. It's perfect. I have two questions. One question more philosophical and one very, very specific. Okay. More philosophical. Exists very big number of work about aging in rats and mice, and we have data for this aging. And also exists a very big count of models about simulation of heart failure, ischemia, and others. I speak about cardiomyocytes model, but no one model of aging, only some specific, for example, KATP currents or something. It's very, very not developed area really. And why this happened? It's my first question. Why this area not developed if we have really data from mice and rats and from animals? And other, I'm very impressed in your presentation part about the neurological activation, because it's very difficult to introduce neuronal ganglia on the heart because we don't know its position. We know that his activity is different for different ages, but we don't know how it really works, and he sends activation in different points and these things. I don't know. I'm sorry, I don't know how to translate it correctly because I don't know the Latin name. And we have a very big problem with acetylcholine expression in this ganglia, because some last results show that exists non-discrete expression of acetylcholine, different variants. It's very dependent from different things which used for animal experiments. For example, any beta blockers also influence on the ganglia, and we don't have a poor result. What to do with this? It's, for my opinion, it's absolutely impossible to.
Okay. Going back to your first question about why don't we have models of aging in the human heart? Well, we have different data in animals. The problem is that most of the effects that are quantified in animals we don't know that they work in humans. In fact, there are differences between species. So this makes us wondering whether human is going to be different, which is probably the case. And also, most of the studies have focused on quantifying differences at the population level. So we do know what happens in animal models at some age and another age, but in mean in the population, so as an average of the population. But we don't have characteristics of inter-individual differences, which we believe is really key, because otherwise you only have a rough idea, but probably won't work when you have data from the patients. So that is one thing that makes things complicated to translate from animals to human. And also in human, the difficulty is to get the data. So it's difficult to get, we have the ECG signals, of course, we have the blood data that are that can be acquired with, rather easily. But what about the cells and tissues? How do they behave in relation to age? And that is difficult. I mean, we are getting those biopsies, but of course, these are from patients. They are not healthy subjects, because otherwise they wouldn't go to surgery and we wouldn't get the data. So you need to do as much as you can with the data you can have. So we get the data from a specific region that is not affected by, but still, this is a limitation, and it's not so easy to get the biopsies in a routine manner. So the data is limited, and this affects the possibility to develop the models to account for that. So that regards the first question. And the second question about the autonomic modulation, of course, yeah. We only attempt to model the sympathetic part. So we for this project, we won't target the vagal modulation, because that would introduce another level of complication. But regarding the sympathetic modulation, there are many uncertainties about how the interconnections with the ventricles are spread along or throughout the ventricle. So that is one difficulty. So what we are doing so far is to take an approximation of what we believe could be something reasonable based on the data we have from the literature. But we don't have any more data to include them in the models. Yeah. Thank you very much.
Yeah, thanks for the presentation. I have a bit of a question, because as you said before, you get your tissue samples and so on from deceased patients, and then you fit your models to the data you acquire, and you're trying to sort of distinguish what is the effect of aging, and so how do you rule out the effect of the disease in your modeling part?
Yeah, that's the main point about using data from patients. What we do is to put as much restrictions on the selection, to the input criteria for the patients to be selected for the study. So they are patients that are going to surgery, but we try to get the biopsies from a region that is not affected by the coronary arteries that may be occluded in the patient. So from any region that is supposed to be close to healthy region, but still we cannot rule out any other effect on the data that we get. So for instance, we do not consider valvular patients or heart failure patients, of course, because the remodeling that we get in the ventricle, of course, would be altering anything that we quantify in the biopsies. So we try to be as restrictive as possible on the data. So we put constraints on terms of left ventricular ejection fraction, ventriculogram data, echocardiographic data, as many constraints as possible, but still, they are patients, they are not healthy subjects.
And the other follow-up is, because you also spoke about intra-subject variability as well as inter-subject variability, but you fit the model to the patient, to your biopsy, and then how do you account for the intra-subject variability as well in your modeling process? Yeah,
what we do is we get the biopsy, and we have several, we divide it into several tissue slices. So we have data from different slices. So, and in each slice, we can quantify data at different regions within the slice. And also we get cellular data from cardiomyocyte isolation. So in that way, we can quantify differences between tissues or between regions within the tissue or between different cells.
And that's how you put your stochastic model then?
And we use our stochastic models for that.
Okay,
yes.
Thank you very much.
Thank you.
I have a question regarding your regulation by stretch.
Oh, okay, yes.
Okay, so you said you included stretch-activated channels. As far as I know, we don't know really the molecular identity of those. So what did you include it? And as far as I understand, you have an electrical model, but the activation will depend on the mechanics. So how do you include this in your action potential then?
We've coupled. I mentioned it, but very briefly. We've coupled maybe I was too quick in saying that, but we coupled the electrophysiology with the beta-adrenergic signaling and the mechanics model. Of course, and it depends on the level of stretch. So we model the stretch-activated channels as having two components, a specific potassium-specific and then non-specific calcium channels, and of course they are dependent on the level of stretch. Yeah,
I missed that
sorry.
part.
Yeah, maybe I was too quick when saying that about the mechanics. Yeah.
Any other questions?
Yeah, I have one. Yeah, it's
sorry.
indeed. Last week I was to a talk, and of people working on cardiomyocyte synergetics,
Okay.
and combining models and experiments. So they found very strong evidences that after cardiac event, the internal flow of calciums within the cardiomyocytes was largely altered by 1-2 orders of magnitudes. And is this, could you through your approach, so go to explore this kind of alterations, and to see, because the issue of this is that while cardiomyocytes were considered as still functional, because of this, then they might not be functional, and then the risk of arrhythmia might be very increased after the first accident.
Yeah, that's very interesting investigation. I mean, we could do something in the models, but what we do so far is to measure both voltage and calcium transients, and that is the data that we use to develop the models, to identify how the model should work to adapt to those data. But we don't consider any other type of approach for that.
So I realize that this may be a bit way further down the road, but let's say you make your model for the aging and you include it in the models. Like, is the end game to sort of fit the model to each patient, and then you can model this patient through time, or is it like making a global model for aging and try to make some general predictions with it?
Well, rather than a global model for aging, a set of models for aging to model the differences between different individuals. Regarding your first comment on whether we plan to follow a patient, this will only be able to, it will only be possible to do it based on ECG data, for instance, because that is the only data that will possibly be acquired through time. So the idea is to get this population of models for aging, for representing inter-individual differences, and then see whether there is something in the ECG that is reflective of what is happening in the electrophysiology of the heart. And if there is something in the ECG, that is something that can be followed a long time.
I think it's time now to move on. So thanks again, Esther.
Thank you very much.